some of the work we've done on dry hopping, specifically West Coast style IPAs. Well, you might be familiar with some of this work too, or you may not be, but when we first came across these New England style IPAs, and they were being described as being very hazy, not extremely bitter, but having massive hop flavor, we wanted to investigate whether these New England style IPAs were really that different from West Coast style IPAs. So we wanted to do a deep dive analysis because we had a very large database already for West Coast style IPAs. So we contacted 12 breweries in New England and told them about this project we wanted to work on. And we were fortunate enough that we got about 12 breweries that were able to send us beers. We told them to send us the beers as soon as they were packaged because we wanted really fresh beers in order to do this analysis. Anyways, one of the reasons I put the hidden secrets in this title is because one of the thoughts I had before we started this project was the haze. I was wondering if the haze was hiding something or possessed some special properties. So with that, we'll get started. All right, trying to get to the next slide here. What's going on? Okay, so what I'll do first is talk briefly about how to brew a New England style IPA. And as I mentioned, we received 12 commercial beers, and we analyzed these using an instrument called high performance liquid chromatography, also known as HPLC. And what we did is we measured various hop compounds that were in these beers, and then we compared the analysis of those hop compounds to what we found in West Coast style IPAs. We also ran a number of tests on the haze itself. We ran some turbidity tests on these beers, yeast counts. We looked at what the hop acids contribute to the haze in beer. We also did a centrifugation test, and then we actually did analysis on the haze itself. And then we also did some haze stability testing. And then some other tests we did on these beers was the IBU test and beer foam. So how to brew a New England IPA? Well, a lot of people believe treating the water with some chloride and some sulfate helps improve the mouthfeel or softness of the beer, as well as the improves some of the hop character. So 150 to 200 ppm of chloride and about half that dose rate in sulfate is usually what's used as a treatment. Although there are a lot of people who believe that maybe this kind of these ingredients really aren't that necessary. The grain bill is also kind of more, I would say, similar to what one would see in a wheat beer. Usually one uses about 10 to 50% of a high protein adjunct like flaked oats or flaked wheat. Just so you know, oats and wheats are very high in protein, with wheat having about 80% of its protein as a very specific protein called prolamins. And for those who don't know, prolamins are haze active proteins, and they're known for complexing with polyphenols to form a fairly stable haze. And the rest of the grain bill is generally just a two-row pale malt. The hop varieties that are usually used in this style of beer are usually citrus varieties or fruit-forward varieties. And the amount of hops added during dry hopping is quite high, usually over 3 pounds per barrel. And there's a lot of brewers who believe that high germinal hop varieties are also ideal for this style of beer. And varieties that are very high in germinal that have been reported in the literature are varieties like Bravo, our experimental hop 09326, Sultana, as well as Cascade and Centennial, just to name a few. Yeast strains are also quite important for brewing this style of beer, kind of like a wheat beer. You know, if you don't use the right yeast strain, you won't get that kind of clove or banana type of flavor in the beer. And it's the same too with these New England style IPAs. Varieties like London Ale 3, Dry English Ale, and Conan, and Safe AL33 are varieties that brewers are using to kind of enhance that kind of juicy, fruity flavor. Now, how the hops are used in brewing a New England IPA is really important. Little to no hops are added to the brew kettle. And so, again, and if there is an addition, it's usually to the whirlpool. And a lot of times the whirlpool temperature has actually been reduced to less than 180 degrees Fahrenheit. And the purpose of that is to not only minimize alpha acid isomerization, but primarily to capture the hop oils and the aroma compounds that are within the hops. How the hops are used during dry hopping is also really important. The hops are actually usually added about 24 hours to 48 hours after the yeast is pitched. And in many cases, the hops are added at about 1 pound per barrel per day for at least 3 days. And the idea behind adding the hops during active fermentation is to take advantage of the biotransformation that can take place. I mentioned in the previous slide that geraniol, hop varieties that are high in geraniol are believed to be important. And it's primarily because, as you can see from this slide, some researchers found that geraniol can be biotransformed into beta-citronellol, into linalool and alpha-terpenol, as well as a bunch of other esters of these hop oil alcohols. And so geraniol-rich hop varieties are believed to be valuable in brewing this style of beer. And as you can see here from the specs of this style of beer, these are pretty big beers with higher alcohol levels, 6.3 to 7.5. The IBUs, again, range from 50 to 70. We actually measured one beer that had as much as 108 IBUs. The color of these beers are usually pretty on the light side. And so we'll move on. So the hop compounds that we tested, again, we tested for the bitter hop compounds like isoalpha acids, humulinones, and alpha acids. And then we also looked at some of the non-bitter compounds to see if they were present in the beer. And these would be beta acids, myrcene, which is a hop oil, and xanthohumol, which is a unique polyphenol found in hops. So as I think most people know, when alpha acids are boiled, they will thermally isomerize to form isoalpha acids. And 1 ppm of isoalpha acids will contribute an organoleptic bitterness of about 1 IBU. Alpha acids are reported to be about 1/10 as bitter as isoalpha acids. And another hop compound that some people are unfamiliar with is humulinones. Humulinones are formed in hops through the oxidation of alpha acids. And humulinones are very similar in molecular structure to isoalpha acids, except for one thing. Humulinones have this hydroxy group present here, whereas isoalpha acids has this hydrogen group. Now, what this hydroxy group does for humulinones is it makes it more polar. So that means it should be more beer soluble and more water soluble. It's also been reported that humulinones are about 66% as bitter as isoalpha acids. And the other thing that we did as a little side project is we took some unhopped beer and we added 12 ppm isoalpha acids to one bottle, and to another bottle we added about 20 ppm of humulinone. And so 20 ppm of humulinone, if it is 66% as bitter as iso, would have an organoleptic bitterness of about 12 IBUs. And so when our panel tasted the two beers, we did find that the two beers did have very similar bitterness intensities. However, the humulinones had a much smoother non-linger bitterness associated with it. And that kind of makes sense. If the humulinones have this hydroxy group, which makes it more, let's say, water soluble and more polar, it would be less likely to stick and linger on the tongue like isoalpha acids. So we do think that this humulinone is absolutely about 66% as bitter as isoalpha acids, but it has a nice smooth non-lingering bitterness. And this could be why a lot of people feel that these New England IPAs have this nice, smooth, bitter flavor. It's because of the humulinones. So the way to measure these compounds accurately in beer is by using an instrument called high-performance liquid chromatography. And this is what a typical instrument looks like. And the way it works, it's quite simple. You have a solvent, which we generally refer to as a mobile phase. It goes through a pump, which pushes the solvent under high pressure. And then we have an injection point. And you can literally inject a tiny amount of your beer into this injection point. And then the mobile phase pushes your beer through a special HPLC column, and it has special packing material. And what that column will do is it'll separate the various hop compounds based on their polarity. And so usually what happens is the first compounds that come off are the more polar or the more water soluble, with the last ones coming off being the least polar and least soluble. And then eventually they'll go through a detector, and that detector will start measuring a bunch of peaks. And then you can measure the concentration of the various hop compounds based on the peak area. In order to use HPLC, you do need an HPLC calibration standard for the various hop compounds, and those are all commercially available. So you can buy an HPLC calibration standard for alpha acids, for isoalpha acids, for humulinones, for beta acids, etc. So this is what an HPLC trace looks like on beer that contains humulinones, alpha acid, isoalpha acids, and alpha acids. As again, for isoalpha acids, we know there's a co and an add, and we can measure all three analogs. And the same thing with alpha acids. We know there's a co, N, and then this little shoulder peak is the add. And the same thing for humulinones. This is the co, the N, and then the add right here. So these can be measured really quite accurately using HPLC. So we measured our 12 New England IPA beers, and you can see here the range for isoalpha acid. The concentration was as low as 5 ppm in one beer, all the way up to 32 ppm, with the average being about 20 ppm. When we look at our database for West Coast style IPAs, the average isoalpha acid concentration is 48 ppm. So as you can see here, the concentration in these New England IPAs is considerably lower. And again, it makes sense. If you're not adding a lot of hop acids to the brew kettle and maybe only doing a whirlpool addition, you shouldn't have a lot of isoalpha acids in these beers. We looked at the humulinone concentration in these beers. And again, the concentration ranged from a low of 12 ppm all the way up to 38 ppm, with the average being about 26 ppm. Now, if we compare that with a West Coast style IPA, you can see the West Coast style IPA only had an average of about 11 ppm. So there's a considerable amount of humulinones, that nice, smooth, non-lingering hop acid in these New England IPAs. What we did here on this slide is we plotted the ratio of humulinones to isoalpha acids, okay? And so essentially what we found is, on average, we had about 1.3 ppm of humulinone for every 1 ppm of isoalpha acids for New England IPAs. So what that means is about half of the bitterness of these New England IPAs is coming from humulinones. And so this kind of helps explain why these beers are known to have this kind of a smooth bitterness, is because about half their bitterness on average is coming from humulinones. Now, if you look at West Coast style IPAs, only about a third of the bitterness is coming from humulinones. So most of the bitterness on West Coast style IPAs are coming from isoalpha acids. Again, we measured the alpha acid concentration in these beers, and the concentrations were actually quite high. On the low side, we saw 17 ppm all the way up to 72 ppm, with the average being about 31 ppm. Again, if we compare that to a West Coast style IPA, the amount of alpha acids in a West Coast style IPA usually averaged at around 13 parts per million. So there's considerably more alpha acids in these New England IPAs. We also looked at the beta acid concentration in these beers, and for me, this was the most surprising and unexpected result, and that was to find actual beta acids in beers at these high concentrations. Again, the low range was 1 ppm, the high was 14 ppm, with the average being about 5 ppm. Just so you know, beer generally does not contain beta acids. Beta acids are extremely nonpolar, so they're not beer soluble. So you almost never, ever see beta acids in beer, but we did see them in every single one of our New England IPAs, again, with the average being 5 ppm. So this was a real surprise. Myrcene, which is a very nonpolar hop oil, we do see in New England IPAs. And again, at concentrations much higher than what we would see in a regular West Coast style IPA. Again, now on the low end range, we saw 0.5 ppm all the way up to 2.5 ppm, with the average being about 1.4. Again, West Coast style IPAs, the amount of myrcene is really quite low. It's usually under 0.3 parts per million. Xanthohumol, this is that unique hop polyphenol that's found in only found in hops, and in these New England IPAs, again, the range was 0.9 ppm up to 3.5 ppm, with the average being 2 ppm. Again, this is versus a West Coast style IPA, where the most we've ever seen is about 0.7 ppm. So this is really quite interesting, because what this tells me is, for those of you who don't know, xanthohumol is an incredibly good hydroxyradical scavenger. And hydroxyradicals are responsible for causing beer to oxidize. So the more xanthohumol you can have in your beer, the better. And so therefore, xanthohumol, in high levels of xanthohumol in New England IPAs, should mean that these beers have incredibly good shelf life. The thing that's surprising, though, is most craft brewers will comment that their New England IPAs don't age well and they oxidize quite quickly. And so that's a little bit odd, given the high levels of xanthohumol. However, one potential explanation could be is the fact that a lot of these New England IPAs are packaged in cans. And if you don't have very good packaging control, you can have beer packaged with high levels of oxygen. I know a lot of the larger breweries like to keep their dissolved oxygen level to below 50 parts per billion. And if you can't obtain packaging levels at that level, then yeah, I could see where you're going to start to have some issues with your beer oxidizing. So you have to have really good packaging. So that could be the cause. So we also monitored the change in some of these hop compound concentrations over time. And as you could see, when we analyzed the fresh beers and then reanalyzed them 3 months later, you could see that the iso concentration just dropped a very small amount, not too much. The humulone concentration actually increased a little bit, but that's probably because if we look at the alpha acid concentration, the alpha concentration dropped a little bit. And again, we know that when alpha acids oxidize, they can get converted into humulinones. So this is probably what's going on here. We see a small drop of beta acids and a small drop in xanthohumol. But again, that could be due to solubility of those compounds in the beer. Maybe they're just kind of falling out. So we measured the haze of these beers using a turbidity meter. And to kind of give you an idea, this is some of the standards you can buy for a turbidity meter. So you can see 5 NTUs. It looks pretty clear, almost water white. You know, at 50, it's just slightly hazy. And then, of course, at 500, it's almost getting almost milky. So as you can see here, when we measured our 12 beers, there was quite a range in terms of haze, with the low being about 119 NTUs and the high end being about 1,700 NTUs, with the average being 547. So these beers were really quite hazy. When we compare that to like a West Coast style APA, usually the haze is quite low, usually about 30 NTUs. So we know with wheat beers, your German wheat beers, yeast can be a pretty big contributor to the haze. And so we wanted to do yeast counts on these beers to see if yeast was also a contributor to this haze. So as a kind of a general rule of thumb, if you have a yeast count of about 1 million yeast cells per milliliter, that'll contribute about 8.5 NTUs of haze. So when we looked at the yeast counts of these beers, as you can see here, almost all the beers except for one beer, this beer J, had a yeast count of below 1 million cells per mils. So that meant for all of these beers, the yeast contributed very, very little to the haze of these beers, except for beer J, where it had a yeast count of about 5 million cells per mil. And it also had a pretty low turbidity as well. So in this particular beer, the yeast contributed almost a third of the haze, but for the rest of the beers, the yeast was very minor when it came to contributing haze. We wanted to see if the hop compounds themselves contributed haze. So what we did here is we took an unhopped beer and added various concentrations of these hop compounds. So again, when we added 3 ppm of xanthohumol, we got almost no increase in haze. When we added 34 ppm of alpha acids, which is quite a bit, the haze only increased about 10 NTUs. When we added 12.8 ppm of beta acids, we did get a pretty big bump in haze, about 30 NTUs, but still 30 is not extremely high. But if you have a combination of all 3 of these hop compounds, you can get your haze to go up as high as about 70 NTUs. And then you're looking at something that's contributing as much as 10% of your turbidity, if not a little bit more, on average. But still, it's not responsible for the majority of the haze, but maybe about 10%. So one of the things we noticed was that these hazy IPAs seem to have high concentrations of these nonpolar or low beer-soluble hop compounds like beta acids and xanthohumol and myrcene. So what we did here is we plotted the concentration of myrcene, xanthohumol, and beta acids of these various beers according to their haze. And so what we found was that when it came to myrcene and xanthohumol, it was actually almost quite linear. The more haze we had, the more xanthohumol and the more myrcene we had. When it came to beta acids, although it wasn't really quite linear, it did follow the same trend. And that is the more haze we have, the more beta acids we have. So what this tells us is it seems like the haze could be acting as a carrier and increasing the solubility of some of these nonpolar flavor compounds. So to see if the haze is indeed acting as a carrier and solubilizing these nonpolar beer compounds, we did this centrifugation test on a couple of beers. And so what we did here is we took 2 beers. Here's our beer 1 is 1H is for hazy, and then 1C is for the centrifuged beer. And again, in the second beer, 2H is for the hazy beer, and 2C is for the centrifuged beer. So as you can see here, the control beer had 1,071 NTUs, and after we centrifuged it, it dropped to just under 300. And in this case, this beer had about 1,700 NTUs, and after we centrifuged it, we lost about half of the haze and dropped down to just under 900 NTUs. So again, if we look at humulinone concentration, now humulinones are very soluble in beer. After we centrifuged the beer, as you can see here, the concentration humulinones hardly drops at all. So what that tells us is the haze isn't helping with the solubility of humanones, and it shouldn't, because it's, again, very soluble in beer. Same thing for isoalpha acids. At these low concentrations, isoalpha acids are very soluble in beer, so there's little to no loss in isoalpha acid concentration as we reduce the haze. Now, alpha acids, again, aren't extremely soluble in beer, and as you can see here, as we reduce the haze, we definitely reduce the concentration of the alpha acids. And we can see the same effect here. As we reduce the haze, the myrcene, which is nonpolar, gets reduced quite a bit. Same thing with xanthohumol. As we reduce the haze, we reduce the xanthohumol. And then again, beta acids. Again, we get a huge drop in beta acid concentration when we reduce the haze. And in this case, because we reduce the haze by about half, reduce the beta acid concentration by about half. So I think this set of experiments clearly demonstrates that the haze is definitely acting like a carrier and increasing the concentration of some of these nonpolar hop compounds. Well, we wanted to do some analysis on the actual haze itself. And since a lot of these beers do tend to develop some precipitate over time, what we ended up doing was decanting a few cans of beer and then collecting the residual precipitate at the bottom of the can. And then we freeze dried that material and isolated the solid protein or solid haze material and sent it out for protein analysis. And like we expected, the haze showed it contained about 35% protein. We were able to analyze it for polyphenols, and we found that it contained about 3.4% polyphenols, and it also contained about 0.9% fatty acids, and these were the 3 fatty acids we were able to analyze for. And the precipitate also contained about 10% starch. And starch is also known to kind of contribute some haze. We looked at the precipitate for hop compounds. We found it to contain about 8% alpha acids and about 3% beta acids, and then just trace amounts of these other hop compounds. Now, as I mentioned, the adjuncts like prolamins, or like wheat and oats that contain prolamins, these rich protein compounds are haze active, and they can hydrogen bond with polyphenols to form monomers, dimers, and trimers. And then these are the hop haze compounds, or these are the, I'm sorry, not the hop haze compounds, but these are the haze compounds that are responsible for these beers. And so one of the problems with these prolamin polyphenols haze reactions is that these compounds, as they form monomers, dimers, trimers, and so on, they get bigger and bigger and bigger, and then eventually the haze molecule becomes so big that it just becomes insoluble and it crashes out. So this is one of the challenges with brewing this style of beer, is that you can brew them hazy, but over time, the haze molecule gets so big that it drops out over time. So some of the tricks some craft brewers are using to try to, let's say, ensure that the beer is hazy when a customer goes to drink them is to, ship the kegs upside down, and to maybe even package the beer in cans so that the cans are kind of upside down. And that way, when, the bar or the customer, goes to drink one of these beers, the keg gets flipped and the can gets flipped, and so that the haze can then get mixed up in the can. Another thing a brewer could potentially do, and they do this in Germany actually quite regularly, is when you buy a wheat beer in Germany, it's a pretty standard operating procedure that the hostess will open up the bottle. She'll pour maybe a third of the beer in the bottle, and then she'll kind of swirl the beer a little bit and then pour the rest of it in to make sure it's hazy. So as I mentioned, we did perform some haze stability of these beers over time. And as you can see, it's very common for the beer to lose the majority of its haze just in the first month. And then over the next subsequent months, the haze continues to drop a little bit more, but you'll lose a majority of that haze in that first month of storage. We also measured the IBUs on these beers. To give you a little background on the IBU test, it really was developed not that long ago. In the 1950s, when Rigby and Bethune made the big discovery that alpha acids thermally isomerize to form isoalpha acids, it didn't take long for some analytical chemists to develop a test method for measuring isoalpha acids in beer. And that test method is the International Bitterness Unit Test, all right, or the IBU test. Now, it's a very simple and straightforward liquid-liquid extraction test method. Again, you take 10 milliliters of beer, 20 milliliters of isooctane, and then 1 drop of octyl alcohol, and then you add 1 milliliters of dilute hydrochloric acid. And you add this to a 50 milliliter centrifuge tube, and then you shake it for about 15 minutes. And what'll happen is after the 15 minutes, you can let the centrifuge settle and let the two phases separate, and you can isolate the isooctane layer that floats at the surface and pour it into a cuvette and measure the absorbance at 275 nanometers. Now what happens is the isoalpha acids in the beer get extracted into the isooctane layer. So when you measure the absorbance at 275 nanometers, that's the absorbance maximum for isoalpha acids. And if you multiply that absorbance by 50, that'll tell you how many parts per million of isoalpha acids is in the beer. And this test method is actually quite accurate if you're measuring a beer that's kettle hopped and contains about 5 to about 35 ppm of isoalpha acids. Once you go below 5 ppm and go above 35 ppm, the IBU and the actual ppm of isoalpha acids will start to skew. Now, one of the things about this IBU test method is that it was designed for measuring only isoalpha acids in beer. As we've just discussed, New England IPAs, as well as regular West Coast IPAs, contain additional hop compounds. They contain hulanones, they contain alpha acids. In the case of New England IPAs, they contain beta acids, and they contain xanthohumols as well as other hop compounds, okay? So when you do this isooctane extraction of New England IPAs, guess what? All these other hop compounds can and do get extracted into the isooctane layer, and guess what? They also can and do absorb at 275 nanometers. So to demonstrate this, what we did is we took some very pure isoalpha acids and very pure hulanones and alpha acids, beta acids, as well as xanthohumol, and we made and we dissolved them into isooctane at concentrations of 30 ppm, okay? And then we ran their UV spectra, okay? And this is the spectra that we get. Now this line right here is at 275 nanometers, right? This is the absorbance, okay, that we get, the absorbance maximum that we get for isoalpha acids. Well, as you can see here, this gray line, this is humulanones, or I'm sorry, this is alpha acids. It also has an absorbance, okay, at 275, and so does humulanone and beta acids, okay? Xanthohumol, as you can see here, has almost no absorbance at 30 ppm at 275 nanometers. So because we know the concentration, because we know the absorbance, we can calculate what's called the response factor. And what the response factor tells us is for every 1 ppm of isoalpha acids, we're going to have an absorbance of 0.7, okay? For every 1 ppm of humulanone, we have an absorbance of 0.6. And for alpha acids, it's 0.6, and for beta acids, it's 0.4. And like I said, for xanthohumol, it's almost nothing, it's 0.07. So what this tells us is, even though humulinones might only be about 2/3 as bitter as isoalpha acids, it absorbs about 85% as much as isoalpha acids. And the same thing with alpha acids. Even though alpha acids are about 1/10 as bitter, they also absorb about 85% as much as isoalpha acids. And even though beta acids are non-bitter, again, they're absorbing about a little over half of what isoalpha acids are doing. So what we did here is we plotted the IBUs versus a very simple formula that we developed called calculated bitterness. And what calculated bitterness is we take the parts per million of these various hop compounds, which we know their concentration because we have an HPLC and we can measure the concentration of these hop compounds very accurately by HPLC. And so we say, okay, we take the ppm of isoalpha acids and we add that to the ppm of humulinones, but we multiply the ppm of humulinones by 0.66 because we know humulinones are 66% as bitter as isoalpha acids. And then we add up the ppm of alpha acids and we multiply it by 0.1 because we know alpha acids are 1/10 as bitter as isoalpha acids. And so we add this all up and we get our calculated bitterness. And so think of the calculated bitterness as really more of your like sensory bitterness. And so what this kind of chart essentially shows us is that the average IBU is about 63 and the average calculated bitterness is about 37. And this is typically what a lot of craft brewers have noticed, is that their sensory bitterness is usually a little over half of what the IBU number would suggest. And this is why. It's because when we do an IBU extraction using isooctane, we're extracting non-bitter alpha beta acids and low bitter humulinones and very low bitter alpha acids. And these are increasing the absorbance and artificially making us believe that the beer is getting more bitter when we dry hop, when actually, in many cases, it really isn't. So again, the reason the IBU test does not work for dry hop beers because in addition to isoalpha acids, humulinones, alpha acids, and beta acids get extracted into the isooctane layer, and they can and do absorb light at 275 nanometers. And because they absorb at different intensities and have different bitterness intensities associated with them, you just can't correlate the IBU test result to the sensory bitterness. And unfortunately, you can't even say, okay, the IBU is going to tell me how many ppm of hop acids I have in there. So the only way you can really get a better sense of what the sensory bitterness is of your beer is to perform high performance liquid chromatography on your beer so you can accurately measure the amount of isoalpha acids, humulinones, and alpha acids. And once again, you have that data, you can plug it into a very simple formula like our calculated bitterness formula, and then calculate the bitterness, and that calculated bitterness will be much closer to the sensory bitterness for that beer. So finally, the last test we performed on these beers was a NEIPA foam stability tester. For those who don't know, the NEIPA stability test is considered the industry standard for measuring foam in beer. And one of the reasons it's well liked in the industry is because the results are very reproducible. Essentially, you could take a can of beer or a bottle of beer, and the beer can be drawn out and then poured into this, this glass. And then there's a flasher, which flashes the beer. And then what it does is one measures the foam collapse over 30 millimeters, and they report the time in seconds. So as you can see here, these New England style IPAs have extremely good foam when they're poured cold. The foam is also very good at 20 degrees centigrade. And the reason we put the results down at 20 degrees centigrade and at 10 degrees centigrade is the test method recommends that you measure the beer foam at room temperature. And the reason they do that is it's a little bit more consistent. One of the things we don't like about measuring beer at room temperature is because beer's generally consumed cold and poured cold. And so we always get much better results from the NEIPA foam test when we do it colder. And we think that's more representative of what the consumer is going to see, as well as the brewer is going to see. So anyways, as a general rule, these beers have excellent beer foam. And again, it makes sense. There's so much protein in these beers from the Haze, and there's so much hop acid in the beer. We know hops and protein combine to make these great beer foam molecules. And so this is one of the reasons these beers have very, very good foam. So in conclusion, the smooth bitterness that many people experience when drinking a New England IPA is due to the high humulone concentration in these beers and the relatively low concentration of isoalpha acids. Like West Coast style IPAs, the sensory bitterness of New England IPAs is a little bit more than half of what the IBU test result would indicate. And again, this is due to the fact that in addition to isoalpha acids, humulinones, alpha acids, and beta acids get extracted into the isooctane layer and absorb and interfere with the IBU test, yet they're only about 2/3, 1/10, and 1/20th as bitter as isoalpha acids. Also, the foam quality of these New England IPAs are very good, and this is due to the high protein content and the high alpha acid concentration in these beers. The haze stability of New England IPA isn't very good, with most beers dropping to less than 400 NTUs within 1 to 2 months after packaging. Analysis of the haze shows that it contains about 36% protein, 3% polyphenols, about 0.9% fatty acids, 10% carbohydrate, and about 12% hop compounds. The use of high protein adjuncts rich in prolamines like wheat are known to combine with polyphenols to form haze. Hop acids can also add to haze, but it's small. And due to the very low yeast counts in most of these beers, yeast is generally not a contributor to haze, but can be at higher concentrations. And finally, the big secret here is that the haze in the New England style IPAs can act as a carrier and can increase the concentration of nonpolar compounds like alpha acids, xanthohumol, beta acids, and most likely other non-polar aroma compounds, and most likely other non-polar flavor compounds which contribute to the unique flavor this beer style has to offer. And finally, I have to thank my colleague, Bob Smith. He's the one who ran all the analysis and tests on these beers. I also would like to thank our New England brewers who contributed these beers, because without them, we couldn't have done this study. And finally, I'd like to thank you for your kind attention. And now we'll open it up to questions.
Yeah, thanks, Dr. May. It looks like we have a couple of questions in the Q&A tab there. The first one goes back to the beginning slide, and it asks, what is the large peak on the first chromatograph slide?
Okay, let me back that up. I'm gonna, I'll minimize this here.
Yeah.
Hold on a second. Okay, now I'll go through them all, but perhaps you're thinking about this right here. This, if that's the peak you're referring to, this is what we call the solvent front. So like I said, there's a lot of water in beer. So this is most likely just water that's coming off. And then again, these three peaks here are the humulinone. And then again, these three peaks are the isoalpha acids. And then again, or the isoalpha acids, and then we have the alpha acids here. So.
Awesome, thank you. Second question, what stage should the hops be added to extract the highest concentration of humulone and xanthohumol?
Yeah. Again, I think what's happening here is, well, I think when it comes to xanthohumol, the haze is trapping it, and we believe the haze is being formed when the hops are being added, incrementally over time during active fermentation. Humanones, on the other hand, are very, very soluble in beer. And so you can make the argument if you wanted to maximize the solubility of humanones in the beer, then you would add the hops perhaps after fermentation, because we do know that the yeast during fermentation is absorbing and removing, some of the hop compounds like humanones. So you probably would be able to get higher concentrations if you're able to do the humanones after. So. Yeah.
Okay, thank you. And next question here. What was the reason that more humanones are in New England IPAs than West Coast IPAs?
Yeah, I think that probably has to do mostly with just the quantity of hops being added, because we've seen some West Coast IPAs with some very high levels of humanones. But usually, again, with West Coast IPAs, usually 1 to 2 pounds per barrel is what they use for dry hopping. But, with these New England IPAs, it almost seems like 3 pounds per barrel in the fermenter or and even after is the minimum. And so, if you're adding 3 to 5 pounds per barrel, you're introducing a lot of humanones to the beer, and that's most likely why the concentrations are higher than West Coast style IPAs.
Yeah. And going back to the HPLC, curious how much of the haze made it into the HPLC? How are samples prepared before the injection?
Yeah. We do sometimes filter the beer before we inject it in the HPLC, but sometimes we'll just inject, with the haze already present. And I'm pretty sure that we did that because we do know that the HPLC column will suffer if you're introducing junk into it. But sometimes for a study like this, we'll sacrifice the column just to ensure we're not, filtering off any, hop compounds. So with this study, we did not filter any of the hazy beers.
Okay. What happens to the beer when you exceed the dry hop saturation point of 8 grams per liter?
Yeah, the dry hop saturation point. That's a good question because I think the dry hop saturation point is dependent on a number of variables. It could be the alcohol content, the gravity, and like we saw in this study, the haze concentration. Like I said, we did a lot of dry hopping experiments making West Coast style IPAs. And like for an example, we could never get the xanthohumol concentration to go above 0.75 parts per million. But in West Coast style IPAs, we're getting, 3.5, 4 parts per million. So this notion that there is a dry hop saturation point, you have to look at what the base beer. There's a lot of factors that can change what that level is.
Mm-hmm. Do you have any recommended mash profiles?
I really don't. Although I guess one would think that, although you have to do a certain period of time for mashing to get good conversion, but it seems like no, I really don't. Sorry. Although actually the one study, it was a paper written a long time ago, and it wasn't for New England IPAs, but it was just for making regular style beer. And it was published, I'm going to guess, oh my gosh, I'm going to guess about, could be even 15, 20 years ago. No, about 10, 12 years ago. And it was by Evan Evans. It's a funny name. He's a researcher down in Tasmania. And he published a paper on single temperature mash infusion. And what he did is he mashed in at 65 degrees centigrade, and he held it there for about an hour. And what he found was that The beer that was produced had less haze, so you wouldn't want to use it, but it had a better conversion than the multi-temperature mashing. And so he had better, just better conversion and lower gravity at the end of fermentation. The gravity was lower. And anyways, I tried it, and because I'm a home brewer, and it's the only way I mash now because I get much better conversion and my beer is a little bit clearer if I'm, shooting for a clear style beer. So that's my only comment.
Yeah,
on that topic.
Thank you. Next question here. Is 10% of Haze contribution from starch carbohydrates or due to residual sugars from fermentation?
Yeah, that's a good question. A lot of these New England IPAs are very high in gravity, even at the end of fermentation. So it could be that. And also, again, using your oats and wheat, you could be getting starch from those, adjuncts if they're not being properly mashed.
Okay. Thank you. And then addressing hop burn, what do you think might be the cause of it? Is it excessive bitterness or could it be tied to a more specific hop compound?
I'm sorry, I'm not familiar with hop burn. Is that maybe a term used for like too much vegetative flavor? I don't know. I'm quickly Googling it now to see if I can find the term, but.
Yeah, and keep in mind all questions can be addressed at a later point too. Yeah, hop burn is the burning feeling you get when you drink a highly hopped New England IPA.
Yeah. My only maybe comment on that is it could be due to a lot of the excessive oil that's in the beer. There's a beer I only get to drink when I'm out in Yakima called Tricytera hops or something like that. So it's like a double IPA, and that's one of the few beers I'll get that sensation where it just, it's like sucking on a hop cone. It's just very resinousy, and I think again it's due to not only a lot of hop acid in there, but I think a lot of hop essential oils as well.
Yeah. And looking at biotransformation, you mentioned 24 to 48 hours after pitching yeast. Would this be different than adding hops at knockout?
I personally like the idea of adding hops like at knockout. One of the reasons is you are allowing, of course, a lot of those hop oils to enter fermentation from the very beginning. And you do get, again, some additional biotransformation. The other thing too is when you add hops, and again, this is really dependent on the hop variety. Some hop varieties are actually quite high in glycosides, and glycosides are sugar molecules which are bonded to essential oils like linalool glycoside or geranol glycoside. And so if you have a hop variety, and it's kind of funny because there's some varieties that, and there's been papers published on it. I can't think of any offhand, although I've got them saved, and I could forward them to you. But there's some varieties that are rich in like linalool glycosides, and so those are the ones you want to be added on the hot side, because if you add them on the cold side, they're not liberated. So the heat will liberate and free up those linalool glycosides, just like there are some hop varieties that are actually very high in linalool and have very low linalool glycosides. Those would actually benefit from adding on the cool side. And so based on some of the research that's out there on those varieties that are, let's say they call them free hop oils and then bound, because they're glucoside bound, we want the bound ones to be added to the hot side and the cool and the free ones to be added on the cool side.
Yeah, awesome. Good conversations here. Great questions. Next one here. Is there an ideal amount of wheat or oats to be added due to their differences in protein? Is one preferred to the other, or does a beer benefit from both?
You know, I think the beers do benefit from both. I have heard some craft brewers say they get some of their best foam when they're using about maybe 7 to 10% wheat, but I don't recall offhand what their oat contribution was. But my understanding is they still need to add some oats too, but maybe not as much as the wheat.
Okay. Next question here. If haze is the carrier of the majority of the percentage of the aromatic compounds and the stability of the haze, how could we expect sensory profiles to be stable?
Yeah, that's exactly true. As you start to lose your haze, you'll be pulling out some of those flavor compounds just because, like I said, the haze is keeping them in solution. That's why, and unfortunately, we never did any sensory on this, but it's certainly something that you guys, out there can do. And that is, again, as I had mentioned before, the way they do it in Europe is they pour out about a third of the beer and swirl it around and then pour out the rest. So whereas if you just open up a can of beer and you let's say decant it without making sure it all goes in, you may be leaving some of those flavor compounds behind in the precipitate. If you're kegging beer, then you could be stuck because, like I said, if the material falls out of the keg and the beer comes out kind of clear, yeah, you could be losing a lot of flavor that's just bound to the haze.
Yeah.
And I was going to say, next week's talk, which Darren's going to talk about, is focused specifically on the flavor compounds and the aroma compounds of New England IPAs. So my colleague Martin Bendel will be talking about that specifically next week, next Tuesday.
Yes, thank you for mentioning that. Yeah, the hop aroma champion from Martin Bendel. Next week, same time here. Please tune in. Yeah, we have time for a couple more questions, right?
Yeah.
Yeah, so are humulones present in CO2 extract?
No, not really. I did see some CO2 hop extract that had very, very small concentrations, but it's small. But the majority of it is left behind in the spent hop material. So the spent hop material is the leaf material after we do the CO2 extraction. The other thing that's interesting too is xanthohumol is also not very soluble in CO2. So the majority of it too is behind in the spent leaf hop material.
Okay, awesome, thank you. In canning with lower levels of oxygen, lower than 150 parts per billion, will it improve shelf life of a New England IPA? This will be the last question here. And if we did not get to your questions, I apologize, but we will definitely take note and then send a response at a later time.
Yeah, I was going to say on the very last slide here, maybe I'll go to that right now. I should have probably gone to that. Yeah, people can see that. Oh, here we go. Or I'll go here. There's my contact information if people want to, email me their specific question if I don't get to it. But the last question, I'm sorry, again?
Will canning at less than 150 parts per billion increase shelf life of a New England IPA?
Yeah, it definitely will. I gave this talk at a conference once, and a fellow came up to me who said he had seen my talk before, and he said that they did improve on their canning, and he felt it really needed to be below 50 parts per billion, but so 50. And he said it greatly, it tripled the shelf life of his beer. So anything you can do to improve on that is the way to go.
Awesome. Cool. Thank you. And last slide there again, Dr. May.
All right. Yeah, so this one here.
Yeah. And that about wraps it up for our first webinar series of Hoppy Topics. Our next presentation will be right here at the same time next week with Dr. Martin Bindle. And our Hoppy topic will be New England IPAs, the Hop Aroma Champion. As always, further discussion is always encouraged. If more questions or comments come up, please visit our website or send us an email. Once again, my name is Darren and I am your host. Thanks for joining us in Hoppy Brewing. Cheers, everyone.